No problem. So next up, we have Lieuwe from Amsterdam University Medical Center. He's a clinical scientist working in Amsterdam, where he's training as a pulmonologist and a principal investigator in an intensive care unit. He co-chairs the Laboratory of Experimental Intensive Care and Anesthesiology and leads a research group who aim to identify effective personalized treatments for ARDS. He has published approximately 200 PubMed indexed articles and received the ERS Gold Medal for ARDS research in 2022. So over to you. Please feel free to share your screen. We look forward to your talk.
I think you see it now.
Yep, I can see it. That's perfect. Thanks.
Thank you. Thank you for giving me the opportunity to present our work at this meeting. I'm sorry, I've just joined, so I've not seen all the presentations, and there might be some overlap in the topics I'm discussing. So I'm going to talk about exhaled breath analysis for the diagnosis of ARDS. Acute respiratory distress syndrome is a acute disease that's mainly seen on the intensive care unit, characterized by protein-rich pulmonary edema, here depicted in a CT scan of the lungs as the white mass that you see throughout both lungs. And the main problem with these patients is that they have difficulty to breathe and require intubation and invasive mechanical ventilation in order to survive. However, it is quite difficult to recognize, and we desperately need bedside testing to diagnose the condition and also to phenotype these patients. First, I will tell you a little bit about the causes and pathophysiology of ARDS, and then bring it back to exhaled breath analysis, where I will talk about previous findings and novel results. So the causes for ARDS are various. It can be caused by indirect lung injury. So for example, a patient has pancreatitis or has an abdominal sepsis, and this primes the endothelial cells to be more permeable, resulting in pulmonary edema. But it can also result from direct lung injury, for example, pneumonia, where the pneumonia first injures part of the lung, which results in a reaction giving permeability to all of the lung, resulting in pulmonary edema rather than just the infectious material of the pneumonia itself. So it's a disease of the alveoli of the lung, where gas exchange is the most important purpose of that area. And if we look into more detail into the alveolar area of the lung, one can recognize that there is endothelium here in the bottom, separated from epithelial here in the top, where gas is exchanged between the alveolar space and the blood in the capillary. And normally the distance between the capillary surface and the alveolar space is kept as small as possible to allow for exchange of gases as smoothly as possible. And of course, this is important in breath analysis as well. And that's why we can measure VOCs that originate from the systemic circulation and that enter the alveolar space through this mechanism. And there are several important biological mechanisms to keep the airspace dry. One of the most important, the sodium transporters within the type 2 epithelial cells. So normally the airspace is dry, there is surfactant on the epithelial side. The glycocalyx is quite important to keep the immune cells from attacking the epithelial and the endothelial cells. And there is little to no inflammatory response within the alveolar surface. Once the alveolar capillary barrier gets injured, there is a leak from fluid from the plasma towards the alveolar space. This contains proteins, including cytokines and adhesion molecules. And these injure the epithelial cells. They disrupt the barrier in between the endothelial cells and cause oxidative stress on the epithelial side. This can, of course, further into a severe injury of the alveolar capillary membrane, in the end resulting in so-called hyaline membranes, which are basically resultants from clotting within the alveolar space. So heavily injured lung with diffuse alveolar damage. So I just spoke about the different disease processes that can be observed in patients with ARDS, summarized here on the right side. And we're getting more and more precise in identifying these pathophysiological processes. Yet, at the same time, we diagnose ARDS with a very simple clinical definition. It should be within 1 week of a known cause for ARDS, for example, sepsis or pneumonia. We look at the chest image to see if there are bilateral opacities consistent with pulmonary edema, and those should not be caused by cardiac failure or fluid overload, and there should be problems with oxygenation. So there is a huge gap between what we know about the biology of this syndrome and what we recognize in the clinic. When we look at such a patient and we want to learn more about the local host response, we should also recognize that this is very difficult to reach. We could, of course, draw blood or take urine, but this is uninformative about most of the disease processes within the lung, although it does inform us about the systemic abnormalities that might have caused the lung injury. We can take bronchoalveolar lavage fluid, but this is quite an invasive procedure that can be done maybe once every few days, but certainly not more frequently. And of course, there is a big interest in analyzing exhaled breath, and we've been working on this topic for this reason for the last 10 years. The most promising feature of exhaled breath analysis is that it could provide results quite quickly. And of course, in an intensive care unit, time is our biggest enemy, and we need results within a couple of hours, making breath analysis quite attractive from that point of view. So, of course, exhaled breath, we can talk about different compounds, but in general, when we talk about intensive care patients and ARDS, we are not interested in the largest portion of the exhaled breath, but just in some of the metabolites that might be present in a tiny, tiny fraction of the exhaled breath. And again, this is maybe also one of our biggest enemies in this field, because we have a hostile matrix to take care of. There is so much other stuff that we are not interested in that it's quite difficult to see a picture of signal within all the noise. I always like to illustrate that with this slide. Here you see 1 part per million in an exhaled breath or in any circumstance. It's almost unrecognizable. Only when we talk about 10 parts per million or 100 parts per million, we can start to see the picture. And this is one of the challenges that we face when analyzing breath samples, especially in the critical care environment, because it means that we need to concentrate the sample in order to see a picture. We also know that the organic compounds found in exhaled breath do not only originate from local cellular metabolism, but can have many origins. They can represent, for example, the host response within the lungs, but they can also come from the inhaled air. Fortunately, this is not a big problem in ICU patients, as we control the air that the patient inhales, and it generally is medical grade gas that is very, very clean. Although some of the modern ventilators do not use gas anymore, but have a concentrator, and this can influence the inhaled fraction of the air as well. There is also a component of bacteria producing VOCs, and in specific indications, this can be of interest, for example, in the diagnosis of pneumonia. And of course, many of the VOCs originate not from the lungs, but from the circulation, although this might be less important in patients with ARDS, where gas exchange in the alveoli is severely hampered. So this is the general workflow that we have had for quite a few years now. We take a breath sample, which can either be analyzed directly or stored, can be analyzed using sensor technology, or in general, we use gas chromatography and mass spectrometry. We use some kind of processing method, a classification method, and evaluate the performance. The most important arrow in this graph is the arrow on the left, where we apply external validation. So use this classifier on a new patient cohort and evaluate how well the test performs. And I'll come back to a few instances of external validation in the examples that I'm going to give now. So all the examples that I will talk about are about gas chromatography and mass spectrometry. Given the audience here, I will not go into detail how this works exactly, but I will come back to this slide to discuss some improvements that can be made to the setup. So first, I will talk about a discovery and validation study that we did about 10 years ago in the University Medical Center in Amsterdam. In this study, we screened consecutive patients on invasive mechanical ventilation and analyzed the breath signals if they were not previously ventilated and had a risk for ARDS. So they were not with completely healthy lungs. And we split that population into a training and a validation cohort based on the time the patient presented to the unit. So it's a temporal validation cohort. And you see on the right side the flow of patients, and about half of the patients in these studies had ARDS. So that's quite a high prevalence. Simultaneously, we try to optimize the sampling approach to allow for frequent and non-invasive sampling in these patients with a method that could be used in clinical practice in the future. And we use the heat-moist exchanger for that. It's a device that traps the exhaled breath condensate of the patient onto a filter. And there's a small pipe in there that you can use for gas sampling, typically for CO2 monitoring. And we validated that in an earlier study against sampling through a side stream connector, which showed similar breath patterns. This was connected through a PTFE tube onto a solvent tube and a breath gas sampler with a mass flow controller that sampled at 200 milliliters per minute. And because these patients are ventilated with positive pressure ventilation, it should be quick responding and not susceptible to changes in the positive pressure during inspiration and expiration. So quite a dedicated setup that could allow for sampling of many patients without disturbing the clinical workflow. In this study, we compared the patients with ARDS to the patients without ARDS in the derivation cohort and identified 3 markers that were associated with the diagnosis of ARDS. Acetaldehyde, 3-methyl-heptane, and octane were all present in higher concentrations in patients with ARDS. And of these, octane was by far the most important marker. Octane, we all know, of course, from the petrol industry, but I should emphasize that these patients were not walking around, so they were not exposed to petrol gases. And in these patients, octane was probably results from the lipid peroxidation of oleic acid, which has been shown to be a predominant feature of patients with ARDS. When we then evaluated the diagnostic accuracy of these 3 metabolites in the training cohort, in the validation cohort, and in a second sample taken from the same patient, a reproducibility cohort, we found that the area under the receiver operating curve was around 0.8, which is considered to be reasonable for this population. And more importantly, we could combine the exhale breath information with the lung injury prediction score. This is a clinical classifier that helps you to determine the a priori risk of a patient for ARDS. And they both contained information that, when added together, gave an area under the curve of 0.91. So we were quite pleasant with this result, as it was around 10% better than most of the plasma tests that we had been evaluating in that same study. And we carried it forward for a validation study. So as an interim conclusion, ARDS could be accurately discriminated with breath concentrations of octane, acetaldehyde, and 3-methylheptane. And when combined with the clinical classifier LIPS, it was better than either alone. But at this moment, we did not have a clinical bed test, but it was gas chromatography and mass spectrometry, something we could not operate at the capacity to allow for a result within 2 or 3 hours after admission to the ICU. We therefore set on to adjust this setup together with people from Philips Research to come up with a model that was easier to use, did not require the helium carrier, was miniaturized, and did not rely on the vacuum of a mass spectrometer to come to a cheaper product. And you can see here that we chose in the end for a room air supply from still from a bottle, a miniaturized flow controller, a simple sample injector, a column oven that was also smaller than the one we used previously, a PID instead of a mass spectrometer, and software for automated peak detection and automated integration of the signal. Next, we tested whether this was sufficient to allow for octane detection in the range that was required. And patients with ARDS had around between 1 and 2 ppb octane, while the patients without ARDS had about half the concentration. And we here showed that in the fit of the calibration gases, we could accurately detect these concentrations within multiple replicates. Subsequently, we used this method to analyze patients undergoing invasive mechanical ventilation without looking at their exhaled breath, without looking at their ARDS diagnosis. And we took 2 samples to study repeatability. And you can here appreciate that the repeatability was quite good, with high correlations and little bias between the 2 samples. We therefore felt confident enough to start a validation study with help of Health Holland and the Dutch Lung Foundation in 2 university medical centers in the Netherlands. And we expected to enroll about 500 patients, of whom 25% would have ARDS. And in this study, in the end, we had exhaled breath samples available in more than 95% of the patients. We had chest X-rays and lung ultrasounds to get to a good reference standard in more than 90%. And even better, we had chest CTs in more than 50% of these patients. The study procedures were as follows. The patients was included as soon as possible after the start of invasive mechanical ventilation. The patient was sampled on day 1 and 2. So the first 2 days of mechanical ventilation using plasma exhaled breath blood gases, ventilation data, and imaging with chest X-ray and lung ultrasound. And then depending on whether the patient had ARDS, yes or no, we continued sampling or stopped sampling. We used the same breath sampling approach as in the previous study because changing the breath sampling approach would also probably change the exhaled breath concentrations and might have impacted the result. Also, this approach was already used quite often in the units and was easy to apply. So was carried forward in this study. And we used two index tests, compact GC, so that is the bedside method that I just described, just for octane detection, and gas chromatography and mass spectrometry, both for octane detection and for untargeted analysis. Furthermore, we spent a bit more time on getting a better reference standard for ARDS diagnosis. I mentioned previously in the introduction that this diagnosis is sometimes subjective. Therefore, we took 3 raters, each of whom scored the presence or absence of ARDS on an 8-point scale. And then we classified patients as certainly having ARDS, certainly no ARDS, or an uncertain diagnosis. When patients had an uncertain diagnosis, they were carried forward into a consensus meeting to allow for a as optimal reference standard as possible, given the clinical definition. In these patients, we screened about 1,250 patients. And in the end, included 580 patients, but some did not provide written consent in the end. So of course, the patient is incapacitated at the moment the study starts, so consent needs to be gained after the study has been completed from family or from the patient. So we ended up with 519 patients in the study, 190 with ARDS and 329 without ARDS. There were 10 compact GC measurement failures and 20 GCMS failures. So we had the reference test available in more than 90% of the index tests in more than 90% of the patients. These are the shocking results. When comparing the octane concentration in the point of care test between patients with ARDS and without ARDS, there was no difference whatsoever between the two groups, resulting also in an area under the curve for the ROC of 0.52, and the LIPS still had a moderately good performance of 0.66. And adding the breath test to the lung injury prediction score does not add anything. Of course, this could be because the point of care device was not good enough, even though we validated its performance previously. And therefore, we were glad we also included GCMS samples to evaluate whether this would perform better. But in GCMS analysis, the performance was quite similar, with an area under the curve of 0.53 and also no added value on top of the lung injury prediction score. Even if we looked only at patients who received a CT scan, so had the best reference standard, there were no differences. When we evaluated patients based on their biological profile rather than their imaging profile, there was also no difference between breath octane concentration. So with that, I come to the conclusion that ARDS is still a common and deadly condition, and the local response is understudied due to limited sampling. And exhaled breath metabolomics seemed very promising. But in independent performance of an ARDS breath test, it was underwhelming. And personally, this made me more skeptical of using exhaled breath analysis for the diagnosis of respiratory diseases in general, as we frequently don't take the opportunity to validate our findings as thoroughly as we did in this study. I will happily take any questions, but not before thanking the team. This work was mainly done by Laura Hagens here on the upper right side, Marie Smit up here, and Anon Heinen. And they were responsible for performing the validation study and have done a terrific job in including more than 500 patients in such a hostile environment. Thank you very much. Happy to take questions.
Brilliant. Thank you so much for the talk. Really appreciate it. I think you make a really important point, actually. Very often, the only thing that gets presented is the initial pilot discovery work. Often it's the follow-up validation, it's not done. So I think it's fantastic that you've been able to get the whole way through to running those validation studies, even if the result wasn't what one would have hoped for. I did have one question on the octane as compound itself. So in the initial discovery work, you made the point that the patient isn't inhaling, but is there other potential sources of background? And if so, I'm wondering whether or not you collected system blanks during that discovery work to see if there's significant differences between what was sampled from the patient and then the system blank without the patient attached.
And that's an excellent point. I didn't mention that. Yes. So in the initial work, we took system blanks every week. So you cannot do it as soon as the patient is connected to the ventilator, of course, but we took them before. And octane was not in that system blank. So it could not have originated from the plastics that the patient is exposed to. So that as a source of contamination was highly unlikely. We did not do the weekly evaluation anymore in the validation study, as they were quite consistent. There is a lot of contamination, by the way, with all kinds of plastics that pollute the chromatogram in our system from about 900 seconds retention time. So we actually don't use that part of the gas chromatogram because of that.
Brilliant, thank you. I think I may have missed it. I think you had touched on it. Was there a mechanistic explanation for why the octane may be present as it relates to ARDS?
Yes, so we really were shocked to find that one, because the classic mechanism to get that in human metabolism would be the oxidative or the lipid peroxidation of oleic acid. That's classically described to provide octane. And it happens to be that oleic acid is found in much higher concentrations in patients with ARDS. And lipid peroxidation has been shown to be a consistent feature of patients developing ARDS. So based on biological rationale, we actually had, even though that was not the reason why we looked at octane at all, that was a machine learning approach, we felt we were in a good shape, much better than we frequently are when we find a VOC that we have a hard time explaining how that could originate from human metabolism.
Excellent. And in the validation study, so there was 3 VOCs that were initially identified in the discovery cohort. Acetaldehyde was one. I didn't catch the third one. Were they all tested? Brilliant, thank you. Were they also tested in the validation study as well, or was it just the octane that was functionalized onto the smaller instrument?
Yeah, so with the GCMS, we still tested the other ones. But with the smaller instrument, we didn't tune them for the other 2 compounds, because in subsequent work that I haven't shown, we found that having octane alone would be about as good as having the 3 together.
Brilliant, excellent, thank you. So we're at time now, but thanks again for a wonderful talk. And again, I think it's incredibly important for the field as a whole to carry things through to run these validation studies. So thanks again.